Hi everyone, welcome to my new channel. My name is Vitaly Stiglyankin. I'm a PhD in computer science from Florida International University. I've been working on bioinformatics and artificial intelligence for about 5 years. Recently, I came across to this Cancer Genome Atlas, or TCGA. So TCGA is a huge research initiative that was launched by National Cancer Institute and National Human Genome Research Institute. The project is very new to me, and even though I work in bioinformatics, my work was mostly related to structural molecular analysis and microbiome, so I never knew details about it. And the way how I learn a new concept is that I search on YouTube like the videos about the concept, and then I get the brief overview, right? So I dig into details in the paper later afterwards if I need to, but it really helps to get the big picture in the head before searching into details, right? And so I was looking for the TCGA on YouTube, and surprisingly, I didn't find any videos. So it motivated me to create my own channel. So in the future, if I want to learn something, then if I don't see it on YouTube, I'm going to make a video so it can be helpful for you as well. So the motivation behind TCGA is to study cancer by looking at the genetic code and other information. So they want to create a comprehensive genome atlas to find specific cancer signatures. Because there are multiple types of cancers, each with specific mechanism, TCGA considers them separately. So in total, they generated samples for 33 types of cancers. So what are those samples? Here are at least 4 major types. First is genomics, where they collected whole genome sequencing of cancer cells as well as normal cells. This type of data can tell you if DNA mutations occurred in cancer, and if so, at what frequency. So the second one is epigenomics, which collects DNA methylation data. So rather than looking at the DNA itself, epigenomics have the information on the process that affects and regularize gene expression without changes of underlying DNA sequence. And the next one is transcriptomics, that tells you what genes got transcribed to RNA. And finally, we have proteomics, that tells you what proteins have been translated. There are also more data such as clinical variables, pathological data, radiology, also molecular data, for which I'm not going to go into details. But as you see, this data has many modalities, which is called multi-omics. So integration of all of those data together, what makes this project successful. There are no specific one gene responsible for cancer. It's a complex system, so DNA transcribes to RNA, RNA translates to proteins, and at each of those levels, we can have abnormalities that can create cascades of events leading to cancer. So it's really important to study all of those modalities together. So let's go for a mini tutorial. This is the official website for Cancer Genome Atlas project. So here's the description, and below are the useful links. So for example, we can go to the TCGA computational tools, and here is the list of all the official tools that you can use to analyze data. So let's go back. Here is the main link to access TCGA data. Let's click on it. Okay, my internet is a little bit slow. Okay, we need to accept the terms. So the project was started in 2006, and since then they collected a lot of samples. So here you can see the distribution of which sites they collected samples, so almost every tissue. So we see that the most samples were collected in lungs, then followed by the breast and bone marrow. So to access the data itself, you need to click on exploration right here. So here is where all the data is sitting. So first we see that it's subdivided by cases, which are specific individuals from where they collected samples. So you see that the pie chart of the primary site, whether the project, disease type, and so on. So if we click on genes, there will be analysis insight related to genes. For example, this graph shows what genes mutated the most. So we see that TP53 is the most frequent genes that appeared mutated in diseased cases. So on the left, you can filter it by type of cancer, like breast, brain, kidney, and so on. You can also click on clinical, which gives you the option to filter by the clinical variables. Like maybe you're interested only at female samples, it will filter it for you. Also, the graphs for genes will be updated. So let's go back to cases. So those entries correspond to individual humans from where samples were collected. So if you click on any of those links, you will see this web page, and it will show you what data was collected for this in particular individual. We see that there is a sequence reads, transcriptome, nucleotide variations, and so on, and each of them correspond to the various number of files. We can click on any of those, for example, to sequence reads, and it gets us to the page where we can download them. So basically, we can just click on it and click download. So I need to register and log in. If you follow the login procedure, then you'll be able to get the data. So again, this data is in public domain. Anybody can go ahead, download it, analyze, and get your own insights. Thank you so much for listening to this video. I hope you enjoyed it. And if you like this video, please click subscribe.